Hi everybody. Today you're going to learn about cell communication and signal transduction. There are two main ways cells can communicate with each other, either in short distance or long, far away. So the first one is autocrine. Auto means self, and so cell is actually signaling itself for some kind of response. And then juxtacrine are where cells are in direct contact with each other. And an example is immune cells and also heart cells. So right here, you've got B and T cells in your immune system, and you can see the cell that's infected with whatever the virus, maybe COVID, is attaching directly to a B cell to activate it. So that would be an example of cells directly having contact with the other one. The other way that cells communicate short distance would be what's called paracrine, and that's where they're not touching each other, but they're at a short distance from each other. So this is two neurons, and the chemicals, the signaling chemicals are being released from this presynaptic neuron with exocytosis. And the neurotransmitters, or the ligands, are traveling across a short distance to the target cell, which has a receptor. Cells can also communicate with other cells that are further away. And this would be like endocrine cells from let's say your pancreas are releasing insulin, and then they go through the bloodstream a long distance to other cells. And one thing I forgot to mention was that in short distance, these cells, the cells are the same type of cell, so neuron to neuron, versus in long distance, cells are usually like endocrine cells, so pancreatic cells, and then all your other cells, so different types of cells. So how do these chemicals actually cause response? And that's what's called the signal transduction pathway. So in the next couple of slides, I'm going to put up some pictures, and so I would recommend that you pause the video and draw and label the pictures. So the signal transduction pathway has three different parts. The first part is reception, where the receptor is going to detect a signal coming from outside of the cell. So that's like right here. Here's your little signaling molecule, and here's your receptor. And remember, those two are specific. So a receptor has a shape like the active site of an enzyme, and then your signaling molecule is a specific shape that fits that. And then transduction, which converts the signal to some kind of form that can actually cause the cellular response. And usually this is what's called a cascade. So, and we'll get into some examples later. So this is going to cause this molecule to be activated, which is going to activate this molecule and activate this molecule. And finally, you get your response, which is the third part of signal transduction. And that is whatever the response is that the signal molecule is giving. So, picture. So signaling cascades relay signals from receptors to cell targets. So what they do is they amplify incoming signals, and that results in cell growth. It could result in secretion of molecules or gene expression. So if you look over here, I would pause the video and I would draw this because this is just a really good example that you might see on your test. So you get your first messenger, right? And that would be something like epinephrine, which is an example of adrenaline. And it's going to bind to its receptor, same shape, and that's going to change the shape. So the intracellular part. So intra, remember, means inside. So when that ligand binds to the receptor, this intracellular part's going to change shape, which is going to activate this GTP protein, the G protein, to make GTP, which then, here's your cascade, which is going to activate adenylyl cyclase, and then ATP is going to get turned into CAMP, which is cyclic AMP, which is a secondary messenger, which is then going to cause a response. Okay, so your first messenger is the signaling molecule, but it can't get through the cell membrane, and so it's going to cause, this to get activated, like the last slide, and then ATP finally becomes the secondary messenger cyclic AMP, which is then going to cause a response. And so this can amplify your signal. So remember, amplify means to make bigger, so to get a larger signal. And then also ligands, remember that's your signaling molecule, can open and close gated channels. That's another thing that they do. Okay, move my picture again. Okay, so you have a couple different types of hormones. You can have protein or peptide hormones is what they're called, or steroid hormones, and they're made of different things. Remember, proteins are polar, steroids are a type of lipid, so they're nonpolar. And so if you look at these two, again, I would pause this and draw the pictures. But if you look at these two examples, here is your protein hormone. Here's your protein, and it's in a vesicle, and it's leaving the initial cell, right? So with exocytosis, here are your protein molecules, and they bind to a receptor. And then that receptor causes this cascade. Here's your transduction, causes a secondary messenger to do whatever the response is in the nucleus. So an example of this would be insulin. And what insulin causes to happen is on the actual cell membrane, it causes more glucose channels to be made. So the insulin causes the secondary messenger to go into the nucleus to transcribe the mRNA for glucose channels. So an example of protein or peptide hormone would be insulin. Now, if you look at the steroid hormones, and that would be estrogen and testosterone, again, the hormone is secreted with exocytosis, but notice the difference. That hormone, so estrogen or testosterone, can go straight through the target cell membrane, and that would be because they have the same structure, right? Oops. They are both they're both lipids, so that the hormone can actually make it through the cell membrane and then cause a response or a protein. So that's a huge difference that I want you to make sure you take note of in your notes. So steroids are made of lipids, so they can get through the cell membrane without a secondary messenger. There's no secondary messenger down here in the steroids. Okay, so cells respond to the environment through signal transduction. So this is epinephrine, just like we talked about. So remember, the environment is always changing. And so organisms need to regulate pathways in response to that. They need to change as well to adapt to the environment. So epinephrine or adrenaline causes glycogen to get broken down. And remember, glycogen is just a bunch of glucose molecules that are bonded together, and that's the way we store glucose. So when epinephrine bonds to its receptor, right, it's going to cause this G protein to get activated, which you can see the little arrow, that's going to cause this adenylyl cyclase to get activated. See how that kind of fits? And then that's going to cause ATP to become cyclic AMP, make this protein kinase, and then break down glycogen into glucose. Remember, adrenaline, right, that's what's going to cause your fight or flight response. So when let's say you are hiking at blind day and you come across a mountain lion, and you either need to probably run away as fast as you can or stay and fight, right? That's the fight or flight. And so your body is going to need to break down glycogen to give you enough glucose so that you can respond to that mountain lion. So signaling pathways can also target gene expression, so making proteins. So like the example with insulin making glucose channels, those are proteins. And then apoptosis, which is cell suicide, is also a response of cell signaling. So apoptosis, I think, is fascinating. That's how you got your fingers, right? So in between your fingers right here, you used to have webs. And so apoptosis, those cells were signaled to destroy themselves. It's cell suicide, and that's why you have fingers. The other time that apoptosis happens is if you get a bad sunburn, and then those signals that were damaged really badly are signaled to kill themselves. So when you peel, those cells are giving their lives so that you can survive. Because remember, when we get a bad sunburn, the DNA is damaged, which causes mutations, which can lead to cancer. So again, maybe draw this, because remember, you want to have good notes. All right. Okay, so the signaling transduction can alter cell responses. So if you, like we were saying, if you have mutations, so the mutations can change the pathway. So here you have normal and then you have mutated. And if you notice what's happening here it's all the same, but then the receptor protein is changed. So here's a normal cell, right? The ligand attaches into that and you're all good to go. You get your secondary messenger and then you get your response. But down here, your receptor has been mutated, and so the ligand or the signaling molecule can't bond to it, and so no response happens in that target cell. So remember, a change in the structure is going to change the function. Here is another thing that, having to do with signaling pathways. I don't know if you need to necessarily draw this one, but this is just another example that was actually taken off of the AP classroom. So you might see this. So you have these FGFR proteins. Without the growth factor, they're inactive, and so they're not going to do their response. But once your body secretes these growth factors, then it's activated and it can cause the response in the target cell. The apoptosis is an example of what this protein does. Another thing is that chemicals can inhibit. So back here, we were talking about how chemicals activate the signaling pathway. And now we're going to talk about an example of how chemicals can inhibit the signaling pathway. So again, here's your nerve, here's your target cell, all is good. The neurotransmitters are being released and bonding with their receptor. That's, remember, it's the same shape. And over here, it's inhibited because the neurotransmitters are blocked. So that's how a chemical can inhibit the signaling pathway. And that's it. So have a great day, and hopefully you learned something about cell signaling and signal transduction. If you need to go back over this and make sure your drawings are good or annotate them, you should do that. Have a great day.